Bueno, ¿se oye? Sí. Bueno, pues bienvenidos todos y me da mucho gusto presentar al doctor Denis Thieffry. El doctor Thieffry tiene muchas relaciones con México, de hecho hizo un postdoctorado en Cuernavaca en 95 a 97. Fue entonces cuando yo lo conocí. Y después hizo otros postdoctorados y estuvo en Marsella, en Francia, durante 10 años antes de moverse a la Escuela Normale Supérieure en París y también más recientemente en el Instituto Curie. Y bueno, pues él habla súper bien español, pero habla mejor inglés todavía y entonces va a dar su plática en inglés. So I'll switch into English now. So he's going to talk about reproducible computational modeling of acute promyelocytic leukemia resistance to retinoic acid therapy. Bueno, Denis, si me haces favor, por favor.
Thank you, David. Thank you to all for coming. We are not so many, so don't hesitate to interrupt me, to ask me questions, including in linear. I think from what I understood, you should have the possibility to unmute yourself and then try to interrupt me because I cannot see if you write things on the chat while I'm presenting the slides. So it's a pleasure to be here, to be back here, because I was here 2 years ago for some classes. So I see new faces. So I guess you probably didn't attend to my classes here 2 years ago. The aim of this talk is, in fact, bivalent. There are two parts. One is to present you some recent work that was done by a group of people in France. In fact, I will mention that later, but it's a collaboration between 3 different groups. And one of the key person, in fact, is a Cuban postdoc, but who now lives here in Mexico, and who did this postdoc with us in France for 3 years. The other part that I want to introduce is, in fact, around the problem of reproducibility in modeling studies. So the idea is that I will present you the applications that deal with leukemia and resistance of leukemic patients to treatment, in particular treatment by retinoic acid therapy. But then I will leave some time for some demonstration of software. So there will be 2 parts. So first, I'm going to introduce the biological problem or the biomedical problem. So what is acute promyelocytic leukemia, for short APL? So it's a blood cancer that is characterized by an increase of immature white blood cells known as promyelocytes. Now, in practice, what happens is that people who have this disorder will have some bleeding and clot formation, and this can lead to death, to thrombosis. And the bone marrow gets filled by malignant cells, and then you don't get the functional cells, in particular, helping you to defend yourself against infections. And the good things about this kind of leukemia is that they are really relatively good treatment. It is on one hand based on the elimination of malignant cells, so using some kind of chemotherapy, so killing the cells that are over proliferating, usually together with a transfusion of blood products to minimize the risk of bleeding and thrombosis. And then there is a more specific treatment that use retinoic acid. So retinoic acid, or it's a modified form called ATRA, together with a poison chemical, which is usually arsenic trioxide. In these combinations, in fact, will kill proliferating cells, but at the same time push the undifferentiated cells toward differentiations. And so it helps to maintain at least the differentiation and the development of human cells. So now a few words about the genetic base of this disease. It is most often due to a genetic fusion that involves two genes. On the one hand, a gene called RARA for retinoic acid receptor alpha chain that fuse with different genes. In fact, predominantly, most cases are related to a fusion of the RARA gene with the so-called PML genes for premieloid leukemia gene. Okay, so it's the most frequent fusion. So that accounts for almost 25% of the cases. For a bit less than 5%, it's a fusion involving RARA and PLZF genes. So it's also pro-leukemic genes, but a different one located on a different chromosome. And so what is important to understand is that usually the people who have this fusion have this fusion on one copy. Okay, it involves one copy of these genes. So they still keep some normal version of RARA and PLZF and PML genes, but then they have one copy that is affected by the fusion. Okay. Now, what happens when the fusion occurs? So here I put X RARA to say it could be one or the other, or even sometimes other fusion, but much more rarely, is that the stem cells can lead to some kind of progenitors that are quite proliferating and in fact enable the amplifications of the cell pool. And then this progenitor gets stuck at the level of the so-called promyelocytes, and they cannot differentiate in mature myeloid cells. Okay, so and this goes together with, in fact, the blocking, in fact, of genes that are involved in cell differentiation. So this is well established, and what is also established is that by providing retinoic acid, so retinoic acid is a molecule that is produced naturally in the niche, and that will, in fact, push the cells toward differentiation in the natural or the sane conditions. But in the when retinoic acid is used to cure the disease, then higher dose are used, and in that case, it can relieve the blockade. Here, we get expression of differentiation genes, and cells can differentiate, and that's how the disease can be cured, at least temporarily, okay, during the treatment. Now, the problem is that depending on the fusion, it works more or less well on the long term. So in the case of the PML-RARA fusion, remember, this is the most prevalent case, the most prevalent fusion. The treatment with high doses of retinoic acids plus some chemotherapy lead to at least temporarily relief, but on the long term, only to 90% survival. So 10% of the patients get relapse at some point. And so they usually treat recurrently, and then at some point, for some of the patients, they finally died from leukemia. So this is quite a problem because this is a very prevalent case. It is the most prevalent case, and 10% of the most prevalent case is quite impactful. Now, in the case of the other fusion, the situation is worse because the relapse occur systematically. After some months, the patient gets the leukemia again, and so they need to be treated repeatedly, and finally they die after maybe some months or some years. Okay, this is a less prevalent case, but it's a pretty bad prognostic in that case. Okay, so the idea from the people, in fact, it's a group from Marseille who study this leukemia and try to find alternative treatment, or at least to understand what is happening in the cases of relapse, maybe to design new treatment, was to combine knowledge about the two situations and try to understand what are the underlying mechanisms. Okay, the fusion may be different, so it's not so easy to study directly the most prevalent cases, because it's only the resistance to treatment occur only in a fraction of the cases, but looking at this special more rare fusion situation, we know that the resistance occurs systematically. So if we can understand the mechanism of resistance here, maybe we can transpose some of this knowledge there. Okay. So the biological question that we wanted to address were first to try to decipher the molecular mechanism underlying the apparitions of resistance to retinoic acids combined with arsenic treatment or some other chemotherapy. And then also try to explain the different responses to the treatment depending on the fusion, the genetic background. Okay. And oh, sorry, I think I missed. Yes, no, sorry. So that were the initial questions. And so our colleagues from Marseille, in fact, the group of Estelle Dupré in the Cancer Research Center from Marseille, set up to analyze in vitro, in fact, cells that bear the two different genetic fusions. And so they applied single cell transcriptomic to these cells and tried to isolate resistant cells from the other cells, and then decipher their transcriptome and try to understand what may explain the difference of behavior of the so-called resistant cell versus the other cells. And so the experimental work was achieved by Mathilde Popinot. At that time, she was postdoc. Now she got a permanent position in Marseille. And so the idea, I don't know how familiar you are with single cell approaches. It's not very important to know the details, but the idea is that by separating cells using microfluidic device and then making specific libraries, you are able to characterize which genes are transcribed at the level of single cells. And so you can differentiate the gene expression level between different cells. And usually people will measure the transcriptome of several thousands of cells in a given samples. And these techniques has led to the discovery of a pretty widespread heterogeneity between cells. So here, the idea is that the recurrence of the disease is related to a fraction of the cells that have some specific transcriptional characteristics that could be traced or identified. Okay. So in a nutshell, I'm not going to go in much details of this work that was done by our colleague, but I show you the graphical abstract to give you an idea what were the insights and how we were led to contribute then to try to model and understand this problem. So they had a mice model with the fusion here. They could inject the retinoic acids to mimic the treatment of human patients. Then they collect the cells from the bone marrow. They treat them through this microfluidic device, and then they measure, in fact, not only gene expression, but also some characteristic of the chromatin, the opening of the chromatin in a specific place, which is another way to reveal the activity of some regions. And so this is related to also to the transcriptional activity in these cells. And so what is shown here is a projection in 2 dimensions of this very big space, multigenic space, where each dot represents one cell with some specific gene expression profile. And you see that the different colors are clusters that could be defined using a statistical approach. And the important point is that they were able to identify one cluster that they associate with retinoic acid resistance, okay, by looking at which genes could were activated or not. And so later on, what they could do is to perform downstream bioinformatic analysis. I'm not going to explain that in detail, but the key point there is that they identify one factor called EZH2, which is an epigenetic factor. So it's a factor that contributes to, in that particular case, to repress the expression of many other genes. And this involves the so-called polycomb complex. So they suspect that EZH2 could be important in the appearance of relapse. And later on, they made different biochemical assays using the same cells. On the one hand, they use, because EZH2 is known, in fact, to modify some specific histones associated to the DNA. There was already commercial inhibitor for that activity, the so-called canonical activity of EZH2. And so they test to what extent the use of this inhibitor blocking this activity of the H2 was able to eliminate or not the relapse. And the conclusion was not. It doesn't work. They were still relapse. So they did another experiment, still in vitro. And in that case, they engineer a mice with a deletion of the genes coding for EZH2. And in that case, they could clear the leukemia. So that means that the hypothesis still, because that's a relatively phenomenological observation, the hypothesis was that some activity of the H2 was indeed involved in the apparitions of relapse, but it was not the canonical, the well-known activity here, because the inhibition of this well-known activity didn't eliminate the tumor. So at that point, that's where we come into play. we had more refined questions. So the question became, how EZH2 activities impact on retinoic acid signaling and cell fate decision? In particular, the decision for the cells to continue to proliferate or to differentiate. Proliferation being associated with disease, okay, in that case. And then our aim was to build a mechanistic model that would integrate these different regulatory layers, signaling, because retinoic acid is a signaling molecules, and there are some further signals related to cell differentiation that can be taken into account. Transcriptional regulation, because you may imagine we have several genes involved in cell differentiation that will be transcriptionally regulated and regulating other genes. And this was really original, epigenetic remodeling, which means modifications of histones associated with the genes. In that case, looking more specifically to the polycomb complex and EZH2 activity. And so that's where our former postdoc, José Antonio Sánchez Villanueva, come into play. He worked 1 year in my group in Paris to learn the computational techniques and then moved to Marseille to work in part with Elisabeth Rémy, a mathematician, in fact, in Marseille, and close collaborator of the experimental group led by Estelle Dupré. And so that was the modeling workflow that was used. And then I want to introduce you progressively. On the one hand, we use a tool called GenSim. Maybe some of you already know it, which is a graphical interface that enable you to define regulatory networks and assign logical rules to each of the nodes of the network. And so to perform different types of analysis, considering the network as a Boolean model or an extension thereof, as you will see. GenSim enabled to do various types of analysis. In fact, probably almost everything many things could have been done just with GenSim, but because it is an interactive tool, and I will show it to you later on, there is a problem a little bit to keep track and to ensure the reproducibility of the analysis, because you play with this interactive model, okay? And so also because we wanted to combine analysis with GenSim with the use of other tools, in particular a tool called MABOS that enable to perform stochastic simulation, we use an environment that was in fact developed by a community of people developing and analyzing logical model, and which is called Colomoto for Common Logical Modeling Toolbox. So it's a way to integrate a set of tools that have been developed by different groups. So for this study, we use mainly within Colomoto GenSim itself, because GenSim can be in fact called and used through Colomoto. Another library called BioLQM, it's a Java library, a tool called PyBullNet, and I will explain you a bit more what we could do with this tool, and then the tools that enable the to perform stochastic simulations called MABOS, which is in fact developed also in Paris, but at Institut Curie. And this led us to then make some predictions, define potential targets for future curation strategies. So one thing to have in mind, for sure I cannot go through the complete history of modeling here, but usually when you start to develop a model, it will not work, okay? And so you go from definition of model to different types of analyzing, modifying your model. So there is a loop here, okay? So we had to go back to GenSim, modify the model several times. In fact, José did that until we end up with a model that we think was interesting enough to be published. For sure, it's never the last word, but then we at some point we decided to stop and publish the work. Okay. Any question at this point? No? Okay. Yes. Sure. Because within the collomotor environment, you can use both. But BioLQM is probably a bit more rapid because, but it's also, in fact, BioLQM is some kind of, it's a library, Java library, that is also implemented within GenSim. So it makes sense to directly use BioLQM rather than to use GenSim, but it would give the same results. There are often, and I will come back to that, I think maybe, so first, before, yes, I will come back to that in the next slide. There are various tools within the collomotor environment that enable you to do the same things, okay? So to build a model, we didn't start from nothing. In fact, besides the experimental data that we had at the end, there were already several models out there related to hematopoiesis, not specifically to our problem, but to related problems. In particular, a model published by Krumsek in 2011 on hematopoietic differentiation and myeloid fate commitment and so on. Some models by my group, some models already by the group of Elisabeth Remy. And so we could extract some circuits from this model and integrate them to get our final model here. There was even a model related to APL, but that was in fact quite big, but not addressing specifically your questions. It was not addressing the problem of resistance to treatment and the fusions that we were considering. So we did use some information from this model to generate this, which is the regulatory graph. So the first thing to say is that this model is almost completely Boolean. So all these round-shaped nodes are Boolean devices. So it means that we consider these encoding proteins, or they may also represent in some cases some complex. So for example, the polycomb receptor complex, we will say they are there, or they are absent, or they are active, or they are inactive. Okay, value 1 or 0 in Boolean terms. Now, one node, array, retinoic acid, was defined as a ternary variable. Why? Because I told you that retinoic acids occur naturally in the niche for hematopoietic stem cells, and then for healthy patient lead to cell differentiation, and this occur at let's say physiological concentrations. In the absence of retinoic acids, so that would be the value 0, or for very low value of retinoic acids, even for healthy patients, there would be no differentiation. And so the signals arrive at some concentration and then trigger differentiation. For the disease patient, the treatment involves a much higher, maybe 10 times higher concentration of retinoic acid. So that's why in our logical model, we consider that we can have 3 values, 0, 1, 2, 0 for negligible, 1 for physiological concentration, 2 for pharmacological concentrations, okay? And depending on this value, the array variable will be able to affect some of the targets. So some interactions here in green coming, these are each, are all activation. Some of these interactions will require the level 1, or some others will require the level 2, okay? For the rest, So one important thing is also for people who are not familiar with this graph, you have two types of interactions. Those displayed in green correspond to activations. So it tends to push the target to value 1 when the source is active. And those in red correspond to inhibition. So when the source is active, it tends to push the target to value 0, okay? But you see that some of these nodes receive various interactions, okay? So that means that it will depend on how the different interactions will work together. So this is formally represented by the so-called Boolean rules. So I show you just simple examples. For example, for CCNE2, it's a gene involved in cell cycle control. It depends on 3 genes. In fact, MYC, who is not far away, I don't see it. Here, okay, there's an arrow going up. E2F1 and RB1. And if you look well, RB1 is inhibiting CCNE2. What is it representing? It represents the fact that CCN2 can be activated by MYC or by E2F1, but it is well known, in particular, including in model for cell cycle, that RB, the famous inhibitor of cell cycle, can sequester E2F and in that case impede its activation activity. So this rule can be read as CCNE2 will be activated if there is MYC or if there is E2F, but not RB1, okay, because otherwise RB1 sequester it. So it's a very simple way to formalize this knowledge, which is something that is well established in the literature and also in previous models of cell cycle. Another example here, which is also dependent on E2F, but here you have 3 possible ways to activate. So the vertical bar here is just the pipe sign in computation that here means or. This esperluette, I don't know you call it, ampersand represent the end, and the exclamation mark in red represent the not in our formalism. Okay, so with these 3 different operators, or and not, you can represent whatever logical function. Okay. So for sure, some of the nodes, for example, now if I take MYC, get much more complex rules because they receive a lot of interactions. And it is what the most tricky part, in fact, when you develop a model, is to define properly these rules. And so that's also why there are several iterations going from, you know, the definition of the model to the analysis until you are able to fine-tune some of the rules. We have ways to define default rules, relatively simple default rules, but usually it doesn't completely work. So a typical default rules is to say, I need at least 1 activator in the absence of all inhibitor. So this is a classical default rule that is used usually for logical model, but biology is always a bit more complex. And there are exceptions, so these default rules work in some cases, but not in all. Okay, so not going through all the rules of the model, but this model is available in a model repository with the rules and with proper documentation. What evidence is supporting the different interactions and to some extent also the rules. Okay. Okay, so now we have the model, and we made a lot of preliminary analysis directly using GenSim software. But at some point, we consider that the model is relatively stable. And so now what we want to do is to analyze it in more details and perform a series of analyses in the most reproducible way possible. And for that, as I mentioned, we use this Colomoto environment. So this is the original publication dating back to 2018. And I just want to mention that the way that we can ensure reproducibility is based on, in fact, two components. One is what a so-called Docker container. I don't know. Are you familiar with what is Docker? Docker? Maybe raise your hands if you are not. You are not. Okay. So it's a way to distribute a computing system like a virtual computer that includes, in fact, a Unix core, Unix operating system, together with all the tools that have been encompassed working in the same, let's say, bubble. So provided that you install the Docker application on your computer, and I will show you, I have it on this laptop, then you can take this container, you can import this container and run it. Your computer might be Unix, or it might be Mac OS, or it could be Windows, okay? Docker is supposed to run on all these platforms. For Windows, it's a little bit more complicated because there are so many versions. Depends a bit on which version, and so it's a bit more tricky to get it running properly. For Mac OS and Unix, it's straightforward. In fact, we can also provide the same package, if you want, for people who know what is Conda. So it's Python package. We can do it that way, and that works pretty well for Unix or Linux system, but it's more tricky to get it running properly on Mac or PC. So this is one part, is this virtual machines in which we have all the tools already ready to be executed. And you will understand in a moment why it is important. And then the second thing is something that we imported, in fact, an approach that we imported from data analysis is the so-called notebooks. Who don't know what is an electronic notebooks? Could be RStudio or you all know what is electronic notebooks? Perfect. So here we use a Jupyter, in fact, Python notebook system that is provided together in the virtual environment. So a notebook, in a nutshell, enables you to encode comments or even more complex programs, but in a document in which you also put descriptions and you display the results of the execution of your comments or your code all together in the same electronic document, and which can be easily re-executed if you pass it away, provided that the people have the right environment. So in that case, the Docker container containing the notebook. And I will make a demonstration at the end of the notebook that we published together with the article. Now, what is the Colomoto environment? I mentioned it is the product, in fact, the product of a coordinate effort of many groups involved in the development of tools for the analysis of logical models. And here you see the status just some months ago. I'm not even sure that I know there are maybe a few new tools since then. I think there were 24 then, or over 20. And you see that these tools have different functionalities. A few have some model repository associated, so you can go there and collect some existing model, pre-existing model. Other enable you to modify pre-existing models. Others enable you to do topology analysis, looking for cycles or whatever within the models. Other enables you to make simulations, to look for attractors, stable states, and so on to control it. And you see that all these tools, so each row is a different tool, will have some of these functionalities, but not all. So there is a complementarity between these tools on the one hand, depending what analysis you want to do. And you see also that one given functionality can be covered by various tools. So sometimes, depending on the type of model, it's better, or your own experience, it's better to use one tool than the other. You can also compare the results with different tools. For very complex models, maybe a good strategy, because you are not always sure that what one tool is doing is correct. So you can corroborate the results with those obtained with a different tool. So it provides a lot of potential to perform extensive and diverse analysis for logical model. Okay. So now I go back to our model. Remember, we had maybe I go back briefly just to insist on a few more things. So this is the model that now I am going to focus on. A few additional points. I mentioned array, multi-level variable. It's an input in the model, okay? Because you treat or not your cells, or it receives or not the signal. Now you have two inputs here representing the gene fusion. Why? Because our model will account for three situations. The healthy situation, no fusion. These two inputs then are set to 0, okay? One fusion present, then, for example, the PML RARA fusion present, then this input will be set to 1. Or still the other fusion, then it is this input that we will set to 1. We will never consider these two inputs at the same time set to 1, because this never occurs. Okay, you have one or the other fusion. You don't get the two. And we don't know any patient that have the two fusion. Okay, we could consider this case, but it doesn't correspond to any life experience. There are a few more inputs. Some are always on, like this one. Some may be considered as taking the value 0 or 1. Now, you see that there are many cross-regulation, and that means that a large part of this network is very intertwined and very difficult to disentangle. Okay? So it's very difficult to predict what will happen for one combination of input without using such computational tools. So I go back to the first result. The first things we can do is compute the stable state of the system. So I'm not going to show complete tables because it will be impossible to read. What I show here are selected nodes of the network, and I use always the same color code. In fact, it was already on the network. Some nodes involved in cell proliferation in green, so E2F and MYC, I already mentioned them. Other nodes also related to cell cycle, but rather cell cycle arrest, so impeding cell to cycle in red. And so there is CDKN1, A and B, and TP53, well-known TP53 here. And then some nodes related to differentiation. SP1, CBP alpha and beta, it's one node that represents the two transcription factor, or retinoic acid receptor B. N. Then I have my wild type genes PML and PLZF. N. I have EZH2 and PRC2. Why? Because these are the key epigenetic components involved in the process. Okay, EZH2, I already mentioned it, and PRC2 is the complex that works together with H2 to repress some genes. Okay. Now, in this table, you see that I have 4 rows. The 4 rows correspond to the stable states that are found when my inputs for the inputs themselves are not explicit here because they will be always defined the same way. WT is for wild type. It means no fusion, okay? And you see that for the wild type situation, no fusion, depending on the level of retinoic acid, which is another input, could be 0, 1, and 2. I can have 4 stable states. 2 stable states in the absence of retinoic acid. And one corresponds to the proliferation situation, and the other corresponds to a cell arrest situation. It means that you don't have cell differentiation, but your cell can proliferate or not. And this corresponds to what is known for the progenitors. They may proliferate, but they are not all proliferating at a given time, okay? Now, in the presence of retinoic acid, being 1 or 2 doesn't matter so much. Your cells will differentiate. And you see all these genes here related to differentiation on, okay? So that also is consistent with what is known. The role of retinoic acid at physiological level is sufficient, and if you provide even more, it still works, okay? For the wild type situation. Now, what happens when you have the most prevalent fusion? Remember, that's the one where, in the majority of cases, the disease could be clear using high level of retinoic acid. Now you have something similar for in the absence of retinoic acid, but in the presence of retinoic acid at level 1, you get also a stable state where cell proliferate. It is this one, okay? So that represents the fact that if you don't provide the medication with retinoic acid, you still keep proliferations, okay? Even in the presence of physiological levels of retinoic acid. In the presence of a higher level of retinoic acid, then you have a single stable state that correspond to cell differentiation. So you can clear the disease, okay? You can push the cells to differentiation, provided you have a high level of retinoic acid. So what is not represented here is the small proportion of cases where you will have a relapse. Why? Because we believe, and there are good indications that this relapse, in fact, occur later on because of the appearance of other mutations. So that's something that for the moment is not taken care by the model, okay? Now, third situation or third genetic background is the PLZF RARA fusion. In that case, we get something similar, except in the presence of high level of retinoic acid. And in that situation, we have the possibility to differentiate, but we have also a stable state displaying still proliferation. So that means that for the PLZF RARA fusion, the worst prognostic fusion, we always have a fraction of the cells that can continue to proliferate despite the treatment, okay? So what the model is not telling you is what is the fraction. At this point, at least with this analysis, doesn't tell you how many cells will go in this stable state or is this one, okay? So if we want to explore this further, we need to perform simulations. For a model like the model that we have, computing stable state is very easy. You do it in a snapshot, but performing simulations get more tricky, okay? So we are going to use other tools to do that. So the first thing that we can do is using, and here is getting more technical, so I'm not going to go into the detail, but we can use static approaches that are based really on considering the model and manipulating the model to partition the state space into states that, for example, let's take the first situation where there is no RARA in the wild type. I mentioned that in that situation, you can have some cells proliferating and some cells resting. Okay. So here, what you see in this graph is a partition telling you that among all the possible states for the system, all combination of 0 and 1 for all variable, you have 0.01% of the cells that are committed to proliferate. So for these states, you end up in proliferation necessarily. And 26% of the combinations of the value of the states, I should say, leads to the proliferating, to the arrested cells. So this is proliferation, this is arrested state. And then for large majority, it's not decided yet. It means that both behavior are still possible, okay? And this is because we use the so-called asynchronous updating scheme, where each time several components can change value, we will consider all possibilities, okay? When there is only one stable state, like for the wild type, when you have physiological or high doses of RA, you go to, in that case, to cell differentiation, and all possible states will lead to this stable state, okay? For the PML rara fusion, you have here also choice between two, and here the high level enforced cell differentiation, okay? Note that this percentage cannot be taken directly as percentage of cells that behave like that, but at least it gives you an intuition of, you know, what is favored. So you see that each time that there is a possibility to proliferate, the number of states that lead to proliferation is a tiny fraction, but it's enough to ensure the progeny of the cells, in particular in the disease case. So maybe I will insist more on that case because that's the most interesting one. It is the case where in that PLZF rara fusion, even upon treatment, you have always resistance. What you see is that it's a small percentage, let's say, of the cells, but in fact, it's of the states, the possible states of the model that lead to cell proliferation, or at least that are committed to cell proliferation. But that small percentage may account for the relapse, okay? So this is a very crude, let's say, first view now about the dynamics, the potential of the state space. Or maybe we can partition states between the different attractors. And this is now a way to represent one simulation. So again, we are performing so-called asynchronous simulation, but now we start from one single initial state. And this initial state, in fact, was derived from the literature as resembling the progenitor, the myeloid progenitor cell type. So it is a special configuration of genes on and off, to which we add the PLZF rara fusion on as input, and the retinoic acid set to its highest values too, okay? So what we expect, as we have seen before, in that situation, you may reach either cell differentiation or the proliferative states. And here, what you see is a summary of a big state transition graph where you can go from the initial state to many other states and end up in one of these stable states. In total, in fact, the state transition graph that was built was encompassing over 12,000 states, but we compress it using an algorithm based on the Targen algorithm for the people who know a bit the field of computer science. It's a very classical algorithm, but we compress the graph further so that we capture the key events that is at the basis of the choice between different attractors. And we compress as much as possible the state transition graph so that we can read it. Otherwise, using the classical tangent algorithm, we will have a lot of chains that would make the message much less clear. And what we see here is that we add information about what changes. So here, for example, you have the initial state plus 600 more states that have the potential to go everywhere downstream. But you see that we could, while we do this computation, keep track about what are the key changes to commit to differentiation or to commit to proliferation. And this enables, and here, for example, this involves switching on, the plus mean switching on, this guy, CDKN1A, or switching off E2F1, or switching off MIC1. This will depend exactly which of the states we are considering here. And remember, the differentiation, indeed, state means that we need to switch off the cell cycle related genes, so this guy. Okay, and then other events must occur to reach the final stable state. On the other branch, it's the opposite. We need to switch off some of the key differentiation genes and to switch on at some point, or they need to be switched on already there at that level, cell proliferation genes. And what you see here is just a representation, because you see each of these nodes here now represent, in fact, a group of states. And so here is one representation of just the states in this green node here, where you see that you have a series of, it's not the complete list, but a series of genes that can take the value 1, 0, 8, and so on. And in fact, playing also with the star to compress this a bit, these different columns represent 191 different states. Okay, different combinations of all these genes on and off. Okay, so this gives you a bit more precise view of what are the key events underlying the choice between differentiation and proliferation in that particular case. Now, it's quite cumbersome to make all this simulation, in particular that because the simulation must be done interactively using the GenSim program. This doesn't exist yet, at least in the Colomoto environment. Maybe that's something we could try to implement. But what we can do, including through the Colomoto environment, is stochastic simulation. So what are stochastic simulations? The idea now is to, when you start from the model and you make an asynchronous simulations, you are generate what the so-called state transition graph. So states and possibility to go from states to states using the rules, the logical rules. And this is very similar to the so-called continuous time Markov chain. So provided that we add, we consider up and down rates explicitly, we can use the Gillespie algorithm and apply it to the Boolean models to now have time simulations, have a more physical representation of time. It's not the exact time that we know, but it's let's say more physical time that what we see in the classical logical simulations. And a group at Curie, in fact, the group that hosts me right now, has developed the MABOS package for Markovian Boolean simulation software. that is public and that provide, in fact, it's a C++ software, plus a series of additional utilities that enable to perform these Gillespie simulations based on a rewriting of the logical model. And this rewriting is, in fact, something that we have implemented in GenSim. So from GenSim, you can directly export a model that will be understood by the MABOS package. And this was the work of Laurence Calzone, together with Gauthier Stoll at Institut Curie. They are still developing this package further, in particular to perform, on the one hand, simulations of communities of cells interacting, populations of cells interacting, and also to perform spatial simulations of cells, combining this with the agent-based model. Well, that's something I will not cover, but I will show you some simulation with the original MABOS package. So with the MABOS package, you can produce two types of graphical representations. In fact, it goes first through large tables and so on, but for the biologist, it's much more easy to look at graphical representation. And so here I show you simulation in a very specific case. Again, I use the PLZF RARA situation, the initial states that correspond to the progenitors, and then I will set retinoic acids to level 2. So we are in the bed fusion scenario, the worst fusion scenario, under treatment, okay? What we see here, again, we are not going to follow all genes. Also, the simulation consider all possible genes, all genes of the model. What we display here is a limited number of genes so that we can read the graph. A gene representing proliferation into F1, a gene representing differentiation, SP1, is H2 because it's a special interesting gene for us, because it's the one that we think is involved in the resistance. And then the PLZF-RARA fusion protein, because we have the gene as input, but the protein is downstream and it is considered separately because it can be degraded, okay? And the model integrate mechanism of degradation of this protein. And what we see is that we have this dynamics, which is, in fact, the mean value for each of these variables across time, but compute over typically 1,000 or 10,000 simulations. You have to imagine that each simulation will have like a noisy trajectory. And then here, what we do, what we display is the mean values for each of these value, for each of these variable over time. And that's why you have values moving between 0 and 1, because our variable can take value 0 or 1, but as a mean, some will, at some specific time point, we will have a mix of 0, 1 for each of these variables. And so we are somewhere in between, okay? So what you see here is that at the final state, you get a distribution of cells either differentiating or remaining into proliferation, okay? And we have a percentage associated to that. The percentage may vary a little bit if you redo the simulation, because these are means of stochastic process. So they may, sometimes vary a little bit, but not significantly. The other things that I didn't mention is that to set up the simulation, we need to define up and down rates for each component of the model. But we have no biological data about that. So how do we do? In fact, the first approach is to provide equiprobable rate. Every time there is a possibility to choose between 3 states, for example, we'll say, okay, 1/3 chance to go there, 1/3 chance to go there. And so this amounts to provide the same kinetic rates in some sense up and down to all variables. Okay? If I do that, I get this percentage. So I cannot take this number as in a too much realistic way, because I know my parameters are like, you know, very crude. Okay, I use very crude parameters, but I see that, like we have seen before, that the proportions of cells proliferating, or the proportion of simulation leading to the proliferative state is a minority. Okay, I cannot say, is it 10% or is it 1% or is it 20%? I don't know really, but we see that a minority. What is more interesting is that I can compare these results for the same model with other situations where I make different changes. In particular, now what I want to reproduce are the experiments where I will interfere with AZH2, because as I mentioned in my introduction, AZH2 clearly has a role in, or an impact on the outcome of treatment. So what happens if I block the activity of AZH2, the so-called canonical activity? So it's some of the arrows coming out of AZH2, in fact, playing together with the polycomb receptor on some targets. And what I see is that I have no significant change. Okay, so that means that our model is current with the experimental data that we had, which is that blocking this activity doesn't impede the relapse. Okay, remember that was the experimental validation in the graphical abstract. And now, third situation, I block completely the activity of ZH2, which amounts formally to say ZH2 will be always off. Okay, and so if you look at ZH2 now, it's remain off, is the orange line. It can be on maybe at the beginning, but it will go to zero. Okay, because we switch it off. And in that case, you see that what I got is only differentiation. So again, it's coherent with the experimental data that I showed you at the beginning. Okay, all that for crude parameter setting. But now what we can do is to say, we still don't have any direct means to identify up and down rates. But what we can do is to check how is this behavior of the model that I just showed you sensitive to parameter change. This I can do, okay? So here I show you the example for MIC. And the situation that I just showed you before is the central one. So when I consider all parameters equal, it means that I put as the up and down rates normalized value of 1, okay? In that case, I see I have a minority of cells or simulations that go to the proliferative attractor in greens, and a majority, so one of this bar here show you the proportion of simulations going to proliferation in green, and the rest, majority, to the differentiation in yellow, okay? It's duplicated because I'm going to change this parameter, either the up deactivation rates or the down desactivation rate, okay? So here it's just the same thing mirroring, okay? But now what I do on the right and what I do on the left is either increase the parameter deactivation rates or increase the desactivation rates, okay, by factor of 10, 100, 1000. So pretty strong change in the parameters, okay? And in that case, again, it's also by factor of 10, but dividing by factor of 10, okay? And so what you see here, MIC was chosen because it has an effect. You see that changing the activation rate has very little impact on the outcome. You have, okay, you change it, increasing it, you increase a little bit the proliferation, you decrease it, you decrease a little bit the proliferation. And you know, MYC is a very pro-proliferation gene, okay? But changing activation rate of MYC doesn't make a big difference. This said, decreasing the deactivation rate of MYC does have a much stronger impact, okay? You see the greenery is increasing much more. So we can distinguish what kind of change has an impact, what kind of parameter change has an impact on the system. So we performed that. We did this kind of analysis for all internal component, all important component of the model. Would be really boring to show you one by one each of these parameters. And so we have been working quite a lot to find a representation, a global representation of this, of the impact of parameter change. So here you see on the left what happens when we change activation rates of different genes. So each axis here, from the center to some of the names here, represent playing with the rates for one component, okay? Activation rate on the left, deactivation rate on the right. And you see at the reference is this difference between, so the scale has been chosen in such a way that you see well the difference between proliferation in green and differentiation in yellow. And now what is interesting is where you have big bumps in one direction or the other one. So you see, for example, that the activation rates for PLZF rara proteins is important. To some extent for MYC, we have seen here. But you can contrast that with what's happening for the deactivation rates. So you see that for MYC, for example, the deactivation rate is much more stronger, and it's also important for E2F. So the overlapping of the color is difficult to make it perfect, but you can follow the curves. And that way we can put the finger of key, on key parameter for which the model is sensitive. Okay, so it gives you two important indications. One is, okay, it would be interesting maybe to fine tune these parameters to get more quantitative prediction. But for that, we need the proper experimental data. So this is one thing. The other thing is that we can say, if we know things in terms of intervention, for example, we want to avoid increasing proliferation. We see which component are more sensitive, at least for which there may be more interesting opportunities to intervene. So typically, some of the components related to cell cycle here. Okay, so the slides here are finishing, and then I will go quickly over maybe some of the tools. So the things that we could conclude here is that based on some single cell data and extensive literature revision, including looking at some pre-existing model, we could build a logical model that capture and explain at the same time some key feature of the response of the main subtypes of acute promyelocytic leukemia to treatment, in particular retinoic acid treatment. Just focusing on the stable state analysis, we could recapitulate the phenotypes of differentiated and aberrant, the phenotypes for my different genetic background under different area treatment, and show how these vary depending on the genetic background. The commitment analysis already enable us to, because I didn't show that in details, but the commitment graph that I show you at some point can also be done for different perturbations. And that way, we could already identify some key components that are involved in the decision between differentiation and proliferation. And focusing now more on the perturbation of the H2, the model, in fact, reproduce the experimental results pointing to a non-canonical role of the H2 on relapse. And it add to this, how to say, this experimental fact, because it has been observed experimentally, but it provides a mechanism for that, because the model implement a mechanistic hypothesis that works in our simulations. And finally, the analysis of mutants with different means that I didn't really show in detail here, but I did show the parameter sensitivity analysis. These two types of analysis points to components of the network that are important in the decision, and that it could be targets, maybe in combinations with retinoic acid treatment, to try to improve treatment. So if you want to know more, the paper was published quite a little while ago. And if you want to learn how to use in particular the Colomoto toolbox, we have published very recently a tutorial. And this tutorial is using a different application, in fact, that was published by a different group. It's a paper published by Cizek et al. Quite a little while ago, but that proposed probably the most comprehensive and up-to-date models today for the control of cell fate in mammalian cells. So it may be an interesting tutorial, an interesting model for the community in general. And for this is the acknowledgement. I have cited many of the people on the way. Let me see. Yes, it is 1:20 already. So I guess, I don't know if you have time, and maybe online it's a bit more difficult to follow, but I have here, I can at least show you, maybe not spend too much time because it's already late, but I can show you just how it looks through the tools. So let me, oh yes, I need to do. Yes. Okay. So the first thing I can show you, is that you can have access to GenSim itself and the software on GitHub. So this is the web page on GitHub with some description and so on. This GitHub website also provides collections of models that have been that are stored there directly in the format that can be used by GenSim. And the very last one, I guess, is the one that I just showed you, which is here. Okay. And there you can download the model in the GenSim format. It's called ZGenML. You can also load it in the SBML format, that is a format more general, used by the systems biology community. And you can load a notebook, the notebook that encode all the analysis for which I show you some of the results. So on the other hand, you can go still on GitHub to the web page giving you access to the Colomoto environment. Among different things, you can find the tutorial that I mentioned, and you can even play with this tutorial online, modify, execute, and so on, thanks to the so-called My Binder public website, that enable you to run Docker environment remotely. So it has its limits, but it's a good way to start before trying to install things to be sure that you are happy and that software can be useful for you. And so, among other things, you will find our tutorial there, okay, and can be executed there. Now, what I want to show you is the notebook for this study, but before that, I show you very briefly the GeneSim model. So this is the model shown through the tool GeneSim directly. So it's a Java jar file that you can run, provided you have a virtual recent Java virtual machine on your computer. So it's also multi-platform. And so here it's an editor. So you can move, you can add, you can delete, you can do whatever you want. I can show you if I take one of the gene, I don't remember, I think I show you maybe this. So here I can show you, for example, how you encode the formula that, so the tool enable you to define the network itself. It's a bit like a drawing software, but it's formalizing really the graph. And at the same time, you can define the rules and you can perform many different simulations. I will not go through there, but we have a tutorial for the use of GeneSim also, and we are going to probably soon publish an update. So this is the tool that was used to build the network, define the rules, perform some preliminary analysis until we were happy with the model. Then we switched to the Colomoto environment. And here you see that, so this is the notebook, okay? And you see that the notebook, in fact, is almost an article ready to be published because you can play with the wiki-like style, define title, text, and so on. And I show you here some of the code cells. So this is a first code cells that enable you to import all the modules from the Colomoto environment that are needed for the study. So you see that there is a series of things. You can also load other stuff coming from Python library and so on. Here, what I do in this line is that I import the model directly from the database that is located in Portugal, in fact, for the moment. So if I just to show you that it works indeed, you see I go to this URL and I load the model and then I display it with GeneSim. Okay, so GeneSim is called, it is part of the environment, it is called and it is displayed. Later on, here I use other functionalities, in fact, coming which are part of BioLQM. First, I take the GeneSim model, I convert it in BioLQM, I give it a name, and then I will, using this other Java library, which is also part of GeneSim, but it's a detail, to compute, on the one hand, all attractors of the system, in fact, the so-called trap space, a bit technical, but they are meant, in that case, all to be stable states. So that's something probably more easy to grasp. And in fact, I can execute it to show you that it works pretty fast for this model. So you see, as usual in Python notebook, you have a little star when it's computing, but it looks like a few seconds to compute the trap space. So trap space can be more complex attractors than just stable state, but we have another algorithm to compute the stable state. As the number of trap space and the number of stable state are equal, I can say I have only stable state. I have no other attractors, no cyclic attractors for this system. And then I can compute them for some specific situation. No, in fact, here I compute all the stable state, but I want to show only some of the nodes because otherwise I have 50 plus columns, okay? you see computing the stable state takes a snapshot for this. So I will not go all over because it's online. You can load it and check. But for example, I can display a stable state. The stable state in that format is not very friendly because, in fact, here I've selected only some nodes. But if you want to look at all nodes, you have a very, very big table. And you can, if you want, display the stable state on the network itself. For that, we use GenSim again, okay? But we color the nodes in blue when they're active, in white when they are inactive. So you can decide to visualize your stable state. Yeah, maybe just to show you that for some things you get a bit verbose messages. It depends on the software that are part, but you can always hide this if you just want the final results. But you will see that we have defined many mutant situations. Here you see the MABO simulations for the three situations. So we reproduce the figure that I show you interactively. It takes a bit of time, the MABO simulations, because it's always a few thousand at least simulations for each situation that you want to simulate. And here we have encode a systematic analysis of many mutants for a list of nodes. So you see all the results at once for all the change. Okay. So it's a long list. So this takes time to execute. And finally, I want to show you that we implement with a bit of code, but the computation of the here you see the impact of the changes in the parameters for the parameter associated with each important or relevant genes. And then even the final figure that I show you at the end. It's not shown here. Sorry. So I think I took an old version of the. Oh, maybe it is before. I'm not sure. We did the single perturbation and then we did the integrated. Yeah, it is here. So we put the single after. So here you see that again within the notebook, all the computation to generate this figure is there. Okay. So I stop here and take some question. If you want, maybe online, you can unmute if you have questions. I don't know if there are still people online, but any question about that? Y sí, en español está perfecto. Tired? Yes. So usually, I mean, it will depend. What you call reproducibility. There are many definitions in the literature and so on. For me, reproducibility is when you publish a paper on a computational model, is that if you show some figures, you provide the code or the formal description of the model. Sometimes it's not executable, but it may be a set of equation, a table with parameters and so on. You should be able to reproduce the figures, at least qualitatively, with the equations and the parameter or with the codes. Okay. And I didn't want to insist on that here, but there have been studies showing that for ODE model store in biomodel database, so already some selected models, people who made the effort to submit, and then it has been curated. For this model, only half of the model are fully reproducible. C'est-à-dire for only half of these models, you could reproduce the key results of the paper. This study has not been conducted for logical model, but for my own experience, we are not better. Okay. Typically for very small models, usually results are quite reproducible. That once the model gets more than 5 variables, then. And this is why it's not because people misbehave, but it's because there is always some asynchronicity in writing the paper, making the figures, modifying the code. It's often also the work of several people. And there is also often some lacking information in the description. So all kind of reasons that are not like ill-intentioned, but that it's like for the experimentalists don't do better. It was published in Nature at some point, at least it was published that Nature experimental studies, for Nature experimental studies, a set quite large sets of Nature published experimental studies, only 1/3 could be reproduced by other groups, and only 1/2 could be reproduced by the author themselves. So, but here we could, for experimental studies, we could say, okay, there is variability in biology. Maybe we try to see that, repeat the experiment and so on, but repeating more doesn't work, whatever. Okay, there is a lot of things that are difficult to control. I think for computational biology, we should be able to control everything, but it's not the situation today. And so there is a lot of community debate today about how to ensure reproducibility, because the model become more and more complex. It's more and more expensive to perform simulation and so on. And so we don't want to repeat and to verify everything all the time. But if we cannot trust the work of some other groups, then we are in trouble, right? So there are more quantitative ways to say that something reproducible or not, even when we perform stochastic simulations. For example, we want to be in some margin which is mathematically defined or something like that, but okay. Then it gets more technical. But other questions? Yeah. The algorithm, the original algorithm is a graph compression algorithm, but which is not compressing so much the graph. It's called Tarjan, T-A-R-G-A-N. Tarjan is the name of the author. It's very, very famous in computer science. What we did, so Tarjan algorithm is considering that all the nodes for which it's for directed graph, okay? And so all the nodes that share, that have paths leading to each other, make some kind of equivalent classes from a mathematical point of view, because they have the same reachability potential, okay? And so the original Tarjan algorithm was compressing a given directed graph in a so-called strongly connected component graph, okay? But the strongly connected component graph doesn't compress chains or hierarchical, but no cyclical paths, okay, or set of paths. And so in biology, often you have this kind of chains in the transition graph, and they are not very meaningful. I mean, what is important is to know how many attractors we have, where the decisions are made to reach such or such attractor. So what we did is to revisit the Tarjan algorithm and add more compression. We compress this kind of chains, okay, and we make a more compressed graph. Tarjan algorithm, I don't know exactly how many nodes we would have, but we would have probably of the order of 1,000 of nodes for the one I show you. So it would be very difficult to read, okay? So we compress it much more. Elaboration. Exactly. Yeah, yes, but it's costly. So it's like the Tarjan algorithm. The Tarjan algorithm is costly also because you have to visit every node, okay, in the simulations, at least once. We do it in one visit, but we have to pile more information. And so right now, maybe something we could discuss. I would like to revisit our algorithm because our algorithm is a elegant, I would say, a recursive algorithm. But the problem is that it's making large piles. So when you have a large model, then the recursive algorithm gets stuck. So we have to revisit it, find another way to solve this. We have, yes. What I'm not sure, I mean, we could share it. Not sure you can get access to the code, the original code today, but we have a paper on it describing it, at least theoretically. It's Berengui et al. And we have the code, the Java code. Yeah. Yeah. I will send you the paper. It will be more easy. Yeah. 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 How many papers? Ooh. In the article, you can check how many references we have in the article, but I would get it's of the order of hundreds of paper. I would say maybe a few hundreds, but. I think AI tools should help you to select articles, but it will not help you very much to avoid reading the articles. That's my point of view. Yeah. Yeah. No, I think, I mean, they've been already attempt to generate networks with AI tools directly. And the problem is that you don't know what to trust. It gives you interesting results, but there is some noise and some hallucinations. And the problem is that how can you distinguish them? Either you know already the literature and then you are able to say, oh, this makes no sense. And then you try to trace back and you see it doesn't work. But if you don't know, you are a student and you are discovering the fields, you have to read everything. There is no way you can avoid. Yeah. Yeah, and they use a lot of different wordings and so on. Even the interaction graph, there are two problems with the interaction graphs. One is that it's not always obvious to, depending on the way that the wording is done and so on, to really say who interact with who directly, indirectly, and so on. And then there is a problem of the abstraction. Because for example, I show you the example of RB and E2F acting together on some targets. This is a very specific ways of encoding sequestration, which is, in my opinion, the best way to encode it in a logical model. But how do you say that to artificial intelligence? Yeah. Yes. We get some information about the regulatory function, and we get some information about the dynamics, the properties. And playing with that, we have a loop where we start with default or literature-based rules, look at the behavior, correct the model until it works. Okay, that's how we do. Yeah. Yes. So we don't use directly ISP or in particular ISP. We try to use ISP for such much smaller models, and it was already getting very difficult. So for such a big model, we never tried that. But because the problem usually is very underdetermined, okay? So it's very difficult to have enough dynamical information to constrain enough your model and make ISP work on big models. So that's the problem. To know, if you would know everything about the dynamics, then you would be able to set up the model quite easily. Okay. The problem is that you know only fractions about the dynamics. And so using formal approach as ISP is not very efficient because of that, because the model is so undetermined at the beginning, then you are lost in many solutions. Yeah. So we start from some reasonable default parameterization, if you want, or rules, corrected with what we know from the beginning, and then we see what are the discrepancies, and using real intelligence, we know which nodes are misbehaving, and we correct their rules. Okay, sometimes it is one node doesn't, there is a discrepancy for some nodes, but we have to go upstream to find what to change. But we can do it by natural reasoning. It's difficult to automatize. I don't say it's impossible, but right now, our brain works better than ISP for that. Yeah. No, I have to say, the solution that we propose may not be the final solution. I have always a slide. I don't know if I have it, but I like to show it. I'm not sure I put it in the slideshow. Did I put it? No, I didn't. I have a slide which shows a statement of Box that maybe some of you knows. All models are wrong. All models are wrong. They are oversimplification and so on and so on. And they are based on current knowledge, but some are useful. Okay, so that's Box who say that like quite a while ago. Box is his name, B-O-X. I can also send you, yeah. And in reality, I add something to that. If your model is not reproducible, then you are sure it will be useless at some point. Okay, so I mean, a model is. I consider modeling as a way to formalize reasoning. The experimentalist is doing the same. He is always reasoning and making mistakes, but he does it in a informal way, missing a lot of aspects. When he's facing something very complex, it's very difficult just with intuition to design the best experiments. When you use modeling, you add something to your reasoning that enable you to increase your power. I'm not saying that we can do so much better than experimentalists, but we can prolong its capacity to design experiments and to develop conclusions based on what it knows. For me, it's not more than that. The model is not an end, it's a tool. And it's due to evolve and to change depending of the question. For that reason, for example, I don't believe as to complete cellular models. I think it makes very little sense because it would be so